today we're looking at gene expression. It's GCSE, but if you're an A-level student, I would recommend that you still watch this because the GCSE has changed and it changed after the A-level. So it still makes for a great precursor lesson into A-level. We're going to recap about the structure of chromosomes, how do they fit in terms of chromatids, and when is it not called a chromosome and when is it called a chromosome. We're going to look at what happens in mutations, and we're going to look at how the genes are actually expressed. For teaching purposes, you may see chromosomes appear like this. It's a little bit confusing, especially when you spring onto A-level. So let's have a look at why they would look like this. We know that you'd get one chromosome from mum and you'd get one chromosome from dad. We know that. However, normally in a cell, here's our nucleus, they're all unwound and unravelled. They need to be like that because obviously that's when your messenger RNA is going to get in there, they're going to take the template from the base code. Then they shorten and thicken just prior to mitosis or meiosis. And so that's what this is representing here, a shortened and thickened chromosome. The bit that they've left out is whenever it does shorten and thicken, it also replicates itself. So instead of appearing like this, it would appear as a double strand like this. Okay, so that would be that one. It's shortened and it's thickened and it's duplicated itself. So that's the original, that's the copy. The other one, again, would be exactly the same. Excuse my rubbish diagram. It's shortened and it's thickened and it's also duplicated itself. So that's why they appear like X's. But clearly for teaching purposes, to keep it simple, sometimes they'll appear like this. And when we look at a chromosome, we recognise that on there are regions where genes are. And so you may have the allele for eye colour here, and on a homologous chromosome, a pair of chromosomes, then you would find the equivalent one on the same place, the same locus, the same position. So this is our locus. This is a pair of homologous chromosomes. What makes them homologous chromosomes? The fact that they contain the same alleles in the same position. Now, although this is doubled, this is where we have a new name coming in. So I'm just going to clear this board and then we'll carry on. So my chromosome has shortened and thickened and it's duplicated itself. But this is still called a chromosome. It's just that now it consists of two sister chromatids. Here's a chromatid, and here's a chromatid. And the same from the other side. Remember we said that this might be one that you've got from mum, and this might be one that you've got from dad. It is one chromosome, made of two chromatids. So the whole thing is a chromosome, and each one is a chromatid. So if these chromosomes were in a nucleus, you've still got two chromosomes. However, if we think about your mass of DNA, I'm just clearly making this up, if it was 1 microgram before, now it would be 2 microgram. But our chromosome number is the same. So at GCSE before, we've always just considered that all the genes and all the DNA in a chromosome was useful. And it's our first introduction into GCSE now, with the changes in GCSE, that some of that DNA doesn't code for proteins. So before we might have said this was the gene for eye colour, this was the gene for a hormone, this was the gene for an enzyme, and we considered that the whole of the chromosome coded for useful things, the whole of the DNA coded for useful proteins. However, now what we're considering is that we've got portions of our DNA that don't code for anything. And again, in the past, we used to think that they really didn't code for anything and they were of no use. We used to call them junk DNA. We now call them introns. But they are extremely important because the default position for a chromosome is go, go, go. The whole thing is on accelerate. And so these genes want to be expressed and want to be active all the time. And what our introns do, our non-coding DNA, is they contain start and stop codes. So basically, it's like the brake of a car. So if you've got a gene and part of your non-coding DNA is telling it to be turned off. So perhaps this is a stop region and that is telling this particular enzyme to be turned off. So it's actually a really part of important DNA. It doesn't code for protein, but it's really important. Now, if we have a mutation, a random change that happens in the sequence, the base sequence, that makes this stop part here be turned off, then basically you're going to end up switching this on. And then we've got no control of it. And so what we're beginning to realise as scientists is that these regions that we thought weren't important before are actually really, really important, because a lot of cancers are caused by the stop code being switched off, or a mutation in that, which therefore switches the gene on, which then causes uncontrollable cell division. So recapping on last lesson, one of the things that we said was that 3 nucleotides code for an amino acid. And many amino acids then assemble into a protein. We said that. Now this switching on and switching off of genes is seen, for instance, in organisms that go through a complete metamorphosis. Let's think about the butterfly. It's a caterpillar at one stage and then it's a butterfly. Its DNA is exactly the same. If you take a cell from a caterpillar and a cell from a butterfly, it's exactly the same. However, it's the DNA, it's the genes that are switched on, which is different. And then by switching on those genes, it gives it a different phenotype. So clearly, during that metamorphosis, when it goes from a caterpillar to a butterfly, there's a lot of different switching of genes going on at that time. And remember what makes a protein. It's that long polypeptide chain of amino acids. And the way it folds is instrumental to how the protein behaves, because the chain folds in such a way that lots of weak hydrogen bonds between the amino acids are formed, and they hold that protein in its shape. That is its tertiary structure, its 3D structure. The thing about the stop codes is that it doesn't necessarily stop the code for the whole polypeptide. It might just stop a region of it. Now, if it stops a region of the code, then let's consider this part of our polypeptide chain is not coded for. And that means that that might then fold in a completely different manner. It will have a completely different shape and a completely different function. So the point is, mutations in our coding regions that affect the direct coding of a particular gene is one thing, but also mutations in our non-coding regions, our introns of DNA, are equally important because they are the stop and the start codes for the genes. So what are mutations? They're random and they can just occur by chance. So basically, normally when the DNA is being copied, so the more active a cell is, the more likely there is to be a mutation, because if that cell is rapidly dividing, when it's copying itself, that's when those mutations are likely to happen. Mutations can also happen on a chromosome level. So if we go back to the chromosome that we had before, at mitosis and meiosis, this chromosome needs to separate its 2 sister chromatids. And sometimes that doesn't happen properly, and you can end up with 3 of them, and that is seen in chromosome pair number 21 with Down syndrome, for example. So in other words, mutations can happen in the nucleotides or in the base sequences, or it can happen on a much bigger level, on a chromosome level. Behind me is an example of a mutation that has conferred an advantage. It is the peppered moth, and it comes up in exam papers all the time. Basically, the peppered moth is like a white colour, salt and pepper together, and it's camouflaged against the tree bark in normal countryside, lovely areas that aren't polluted. And that gives it a camouflage against birds that are going to come down and eat it. So lichen, which is the white stuff that grows on trees, if you ever lean against a damp tree trunk, you'll end up with all gunk down your back, and that is lichen, L-I-C-H-E-N. And basically it's an indicator species. So in areas that are unpolluted, you'll find a lot of lichen on tree barks. In more industrial areas, you'll find that the lichen doesn't grow. So when the lichen doesn't grow and you've got a dark tree bark, then this light peppered moth is going to show itself more easily, and it's more likely to be eaten by predators, and it won't pass its genes on. However, what happens is a mutation happens in its coding for its colour. You'll learn more about this in the next couple of lessons, but just to give you a quick idea of how not all mutations are bad. So the mutation happens that codes for melanin, and we end up with a darker moth. It's the same species, it's just got different colouring. And the colouring survives in the population because it confers an advantage. It is now camouflaged against the dark tree bark, because the dark tree bark hasn't got lichen on it. There's no lichen growing because it's an industrial polluted area. And so what happens is the frequency of dark to light moths change depending on what area you're in, because the mutation has given it an advantage. So what are the key points of what we've said today? We've said that your genes are the same, you're born with them, you've got them, but different genes might be switched on or off at different times. And this is really shown when you look at organisms like a butterfly that were once a caterpillar and have had a completely different change of their physical appearance. Their phenotype is completely different, and that's because different genes have been switched on or off. We've also said that non-coding regions don't always do nothing. Non-coding regions of DNA can be start and stop sequences. If you turn off a stop sequence, you're switching the gene on. And this is a massive area of research now for cancer, because don't forget, cancer is uncontrolled cell growth. Remember that mutations are happening continuously. They may happen in non-coding regions, they may happen in coding regions in your genes, but sometimes the changes are so insignificant that it's not expressed in the phenotype, and an enzyme or a hormone is still coded for the same. The other thing to remember is that not all mutations are bad. Sometimes they confer an advantage, and that's going to help the organism or the species. There's a field of study called epigenetics, and you'll study this at A level. And basically, before, we always considered that the genes you passed on to your offspring were just the genes, and the environment had no impact on those at all. What scientists are now finding out is that there are some things that can affect your genes, and that change can be passed on to your offspring. And that is the study of epigenetics.